small, dense, rounded, and granular particles. They contain ribonucleoprotein. They occur either freely in the matrix of the mitochondria, chloroplast, and cytoplasm, or remain attached with the membrane of the endoplasmic reticulum and nucleus. The ribosomes were described by J. E. Floyd in 1952. The name ribosome was coined by R. B. Robert in 1958. The ribosomes occur in both prokaryotic and eukaryotic cells. In the cells in which active protein synthesis takes place, the ribosomes remain attached with the membrane of the endoplasmic reticulum. The cells where such active synthesis happens are pancreatic cells, hepatic cells, osteoblasts, serous cells of submaxillary glands, chip cells of the glandular stomach, thyroid cells, and mammary gland cells. Ribosomes are spheroid structures with a diameter of 150 to 250 angstrom. Each ribosome is composed of two subunits. One subunit is large in size and has dome-like shape. The other ribosomal subunit is smaller in size and it occurs above the larger subunit, forming a cap-like structure. The ribosomes are chemically composed of RNA and proteins. The ribosomal RNA play a central role in the process of protein synthesis. The ribosomal proteins enhance the catalytic function of the RNA. The functioning of RNA is under genetic control. Endoplasmic reticulum. Electron microscopic study of sectioned cells has revealed the presence of a three-dimensional network of sac-like and tubular cavities called cisternae, bounded by a unit inside the cell. Since these structures are concentrated in the endoplasmic portion of the cytoplasm, the entire organization is called endoplasmic reticulum. This name was coined by Porter in 1953. The occurrence of endoplasmic reticulum varies from cell to cell. They are absent in erythrocytes, for example, cells and embryonic cells, egg cells and embryonic cells. Endoplasmic reticulum is the site of specific enzymes controlled by biochemical reactions. Its outer surface carries numerous ribosomes. The presence of ribosomes gives a granular appearance. This condition, endoplasmic reticulum, described as rough endoplasmic reticulum. Rough is the site of synthesis of proteins. Ribosomes are absent on smooth endoplasmic reticulum. Smooth endoplasmic reticulum is concerned with lipid metabolism. Morphologically, endoplasmic reticulum may occur in three forms, namely lamellar form, vascular form, tubular form. Rough endoplasmic reticulum, nucleus, ribosome, and smooth endoplasmic reticulum. Sheet form of endoplasmic reticulum. Lamellar form of cisternae. These are long, flat, sac-like tubules. Their diameter is about 40 to 50 micrometer. The rough endoplasmic reticulum has a synthetic rule. It is mostly seen in cells of pancreas, notochord, and brain. Vesicles. These are oval vascular structures. Their diameter is about 25 to 500 micrometer. They occur in most of the cells. Tubules. These are broad structures forming the reticular system along with cisternae and vesicles. They have a diameter of 50 to 190 micrometer. They occur in almost all cells. Functions. It provides skeletal framework to the cell. Number two, it facilitates exchange of molecules by the process of osmosis, diffusion, and active transport. 3. Enzymes of endoplasmic reticulum control several metabolic activities. They serve as intracellular transporting system. Number 5. It conducts intracellular impulses. Number 6. It helps to form nuclear membrane after cell division. 7. Smooth endoplasmic reticulum synthesizes lipids. Golgi bodies, Golgi apparatus. The Golgi apparatus was discovered by an Italian neurologist, Camillo Golgi, in 1873. The Golgi apparatus occurs in almost all animal cells except red blood cells. Animal cells usually have a single Golgi apparatus. Some cells have more of Golgi apparatus, and most of the ectodermal and endodermal cells it occurs in between the nucleus and the periphery. In neurocells, it occupies a circumnuclear position. The simplest unit of the Golgi apparatus is the cisterna. A cisterna is about 1 micrometer in diameter. It has a membrane bound space. This space accumulates secretions. Numerous such cisternae are associated with each other and appear in a lamellar arrangement. In the lamellar arrangement, the space between each cisterna is 20 to 30 nanometer. A group of these cisternae is called dictosomes. A group of dictosomes constitute the Golgi apparatus. Secretory vesicle, layered cisternae, rough endoplasmic reticulum. Typically, a Golgi apparatus appears as a complex arrangement of intercollecting tubules, vesicles, cisternae. The Golgi apparatus is the site of synthesis of biochemical. They also collect proteins and lipids made in endoplasmic reticulum and add additional substances. Lysosomes. These are tiny vesicles surrounded by a membrane. Lysosomes are involved in intracellular digestion and are primarily meant for destroying unwanted and aged organelles inside the cells. Lysosomes were initially named as perinuclear dense bodies. The name lysosome was coined by C.D. Didop in 1955. Lysosomes occur in all animal cells, however, they are not found in mature mammalian erythrocytes. Muscle cells contain very few lysosomes. They are numerous in epithelial cells of secretory or excretory organs. Each lysosome has a round structure. It is filled with a dense material. Their shapes and densities vary. Their sizes ranges from 0.2 to 5 micrometer. Recent studies reveal that lysosomes may contain up to 40 types of hydrolytic enzymes. The enzymes are mostly proteases, nucleases, glycosidases, liphases, phospholipases, phosphatases, sulfatases. We start digestion, entry of solid matter, lysosome, Golgi apparatus, functionless organelles. Lysosome originate either from the Golgi apparatus or directly from the endoplasmic reticulum. The enzymes they contain are used in dissolution and digestion of redundant structures or damaged macromolecules from within or outside the cell. For example, when an animal cell ingests food into a food vacuole, lysosomes fuse with the vacuole and break down the contents. Their enzymes digest carbohydrates, fat, and proteins. The glands and some digestive organs package their digestive enzymes and lysosomes before releasing them outside the membrane. When a cell dies, its own lysosome release the enzymes that digest the remains of the cell in a process known as autolysis. For oxysomes, these are spherical organelles bounded by a single membrane. They are found in the cells of protozoa, fungi, plants, liver, and kidney of vertebrates. The name peroxisomes was coined by C.D. Dog and P. Baldwin. Peroxisomes contain catalase, an enzyme that catalyzes the decomposition of hydrogen peroxide to the harmless products water and oxygen. Hydrogen peroxide is a byproduct of certain reactions of metabolism. It is potentially a very harmful oxidizing agent. Centrioles. Centrioles are two cylindrical microtubular structures found near the nucleus. When a centriole supports a flagellum or cilium, it is called the vessel body. The centrioles occur in most of the animal cells, algal cells, and some plant cells. They are absent in prokaryotes, red algae, yeast cells, and flowering plants, and some non-flagellated or non-ciliated protozoans. The centrioles range in size from 0.15 to 0.25 micrometer in diameter. They are usually 0.3 to 0.7 micrometer in length. Each centriole and vessel body is formed of 9 triplet microtubules equally spaced around a perimeter. Each microtubule has a diameter of 200 to 260 angstrom. In diameter, the microtubules are made up of a structural protein, tubulin, along with lipid molecules. It was initially considered that new centrioles arise by the division of existing centrioles. This idea is no longer accepted. It happens that new centrioles are produced de novo or synthesized using an existing centriole as a template. In most of the animal cells, centrioles are the focal point for the centrosome. The centrosome organizes cytoplasmic microtubules during interphase in mitosis. It provides the two folds of the mitotic spindle. Centrioles form the basal body and the cilia. In spermatozoon, one centriole gives rise to the cell fiber or flagellum. Centrioles are also involved in ciliary and flagellar activity.